all to the 15th webinar of Plant Breeding webinar series. These webinars are being organized by Foundation for Advanced Training in Plant Breeding on a monthly basis. And today we have Dr. Ramesh Bhat, who will talk about genomic-assisted breeding in groundnut. So the purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmer comprehensions, and various other aspects required for inclusive development of agriculture in order to achieve food security. Foundation for Advanced Training in Plant Breeding, abbreviated as ATPBR, is a non-profit organization. We aim to promote advanced technology in plant breeding and develop qualified plant breeders who would complement and add value to the existing system. Our efforts are towards creating a conducive ecosystem where skillful organizations or resourceful people work together to develop joint courses, research, or co-incubation. ATPBR organized professional training, workshop, and various awareness sessions to build required competencies of the global standard. And we also organize annual conference named as Fiber Farming Conference in different locations across India. Now, I would like to take the privilege to introduce our moderator, Dr. Krishna Prasad. He worked with ICR for about 27 years in various capacities in organizations like CRIDA, IHR, and DOGR. Later, he joined in Adventa as Indian Research Lead, Vegetable Crop, and now presently he is working as Breeding and Research Lead at Tierra Science Private Limited. He has published around 200 research papers and contributed in the development of around 40 varieties and hybrids of various crops including tomatoes, eggplant, cucumber, rich gourd, sponge gourd, pointed gourd, green peas, French bean, pole beans, and delicacy beans. His brinjal varieties are the source of resistance to bacterial wilt in the breeding program all over India. It's really an honor and privilege to have Dr. Krishna Prasad as the moderator. Request Prasad sir to take the floor. Prasad sir, over to you.
Yes, thank you, madam, for nice introduction. Good morning to all. It was a privilege for me to act as a moderator for this program today. I thank you very much, Dr. Aparna Tiwari, for choosing me as a moderator for this program. And today, as a brief introduction, I would like to talk about groundnut breeding. As you know, this is an important oilseed crop, first position in India, followed by soybean. And we produce about 6.75 million tons of oil seeds, the groundnuts, whereas China produces about 17.75 million tons. That is nearly 3 times higher than our production. In 2021, we have about only 49.14 lakh hectares have been sown in 2021. 2022 statistics are yet to receive, and that is less than 1 lakh hectares in 2020. Then in 2020, we sown around nearly 50 lakh hectares. That is nearly 1 lakh hectares have come down in the area of groundnut sowing. And the major states are Gujarat, Rajasthan, Andhra Pradesh, Karnataka, Madhya Pradesh. These are the major states producing the groundnut. And an average yield of about nearly 1650 to 1700 kilograms per hectare. And at the selling price is around 5,200 to 5,500 rupees per quintal in 2021. As we all know, that it is a nutrient-rich food legume, valued for good quality cooking oil, energy, protein-rich food, and nutrient-rich fodder also. And the breeders and the universities and ICR Institute are concentrating for the development of improved varieties for the requirements of farmers, traders, processors, and also consumers. As you know, if you go to US or Europe and all, the groundnut kernel itself is a big size, whereas in India it's a small size. We don't find that much big size of kernels in our country. Maybe due to long day conditions or sink source relationship, we don't know. That may be the reason. But we need to develop such type of gold seeded groundnut varieties may have a more export potential also. Chances are there. Main breeding objectives are enhancement of the yield, tolerance to biotic and abiotic tests, quality traits are the main for breeding program, development of molecular markers, genetic and physical maps, generation explicit sequences. These are some of the techniques which are using in the breeding program, and identification of QTLs for important biotic and abiotic stresses and quality traits are the main turnover in the breeding programs of groundnut breeding program, especially in the abiotic stresses. First, molecular assisted breeding, through molecular assisted breeding, the variety was developed in 2003. And since then, USA, China, and Japan, India are routinely using these molecular breeding techniques and all. And mostly for the drought resistance and drought tolerance and increasing the pod yield. Genotyping by sequencing expected to accelerate the use of genetic tools to enhance the genetic gain for targeted traits in groundnut. And we have none other less than Dr. Ramesh S. Bhat is a groundnut breeder, a proven groundnut breeder, excellent academician, and he is the right person for choosing this, delivering this talk on genomic assisted breeding in groundnut. I introduced Dr. Ramesh Bhat. He is working as a professor in the Department of Biotechnology in US, Dharwad. He had an excellent academic achievements. From BSc AG onwards, he awarded gold medals, and he was flooded with gold medals, even up to PhD level. And he was a visiting scientist for CS Council of Scientific and Industrial Research Organization, Adelaide, and also on Boy Scouts Fellowship. And he was also visiting scientist in USA and also Japan in 2017 and 19. He possessed nearly about 23 years research, academic, and teaching experience. And he has successfully completed some of the 30 research projects, and especially granted by the DBT, DST, ICR, Government of Karnataka, including Indo-Japanese and Indo-UK projects. And it was a great, it's a great achievement to mention that he was granted nearly about 70 million rupees under CAGIR and DST Young Scientist Research Project, fast track project for young scientists. He was awarded nearly 70 million rupees project for this under this category. Since 2009 onwards, Dr. Bhat is working in the groundnut breeding, in especially genomics and molecular breeding. And he developed one in very good and very drought resistant or drought tolerant variety DH256. And this has become common in growers among the Karnataka farmers. And prior to this 2009, he worked in the rice breeding also. He contributed 3 important varieties, which are Sharavati, IET 13901 and KHRS 21. These are very popular among the farmers of the Karnataka. He worked on gene tagging, lectin gene promoter trapping, and lectin gene cloning and transformation. He guided about nearly 23 MSc students and also 10 PhDs in his career, and published around nearly 85 research papers published in both Indian and international journals such as Euphytica, Crop Science, TAG, and etc. With this brilliant academic performance and research performance, I request Dr. Ramesh S. Bhat take over the mic and deliver his talk. Thank you very much.
Yeah, thank you very much, sir, for the nice introduction and kind introduction, rather maybe more weightage given on the introduction. First of all, I would also thank ATPBR, all the members, Dr. Aparna Tiwari, and my teachers also. I could find Dr. Khadi sir, and in fact, I should mention Dr. Khadi's help here, both as teacher as well as the mentor, and as a DR, he always supported my work on Groundnut, and as well as the earlier topic. Yeah. So could you see my slide, sir?
Yes, we can see. Just make it
Yeah.
less.
Okay. And also thanks to all the members, esteemed members of ATPBR and all the members and participants. Today's talk is on genomics-assisted breeding in groundnut. And of course, most of the introduction part has been given already by Dr. Krishna Prasad, sir. In the world, if you look at areas, almost 28 million hectares, and India it is about 5, and Karnataka is about 0.6 million hectares, with a production of 47 million tons worldwide, and in India about 9 million tons, and productivity is also almost 1,600 to 1,700. And if you look at the highest producer or in terms of productivity, it is the USA, and in India, it is the Tamil Nadu state, which produces almost 2,600 kgs per hectare. And importance of groundnut. In fact, Dr. Krishna Prasad has told it's a main oilseed crop next to maybe the first one and next to soybean. Probably it may not be the true case right now, because groundnut nowadays it is more used as the say a food crop rather than the oilseed crop. And it's the fourth most important oilseed crop and predominantly rain-fed crop, and also a cash crop, and grown by resource-poor farmers. And it is an edible oil with an oil content of about 43 to 55%, and food crop very rich in terms of protein to the tune of 25 to 28%. And groundnut cake is a main say food for animals with high protein and used as animal feed. And homs are also rich in protein and more palatable. So it's also used as a fodder crop to a certain extent. And another important role of groundnut in terms of soil health is it helps the soil in building a lot of nitrogen. And globally, over half of the groundnut is going for oil extraction and remaining as raw and processed food. And groundnut is commonly called as poor man's almond because of its rich nutrient content in terms of oil, proteins, fibers, polyphenols, antioxidants, vitamins, and minerals. And it's an excellent source of compounds like resveratrol, where we have a lot of literature now coming in to improve groundnut for this resveratrol, and also a lot of phenolic acids, flavonoids, phytosterols, and a lot of amino acids, almost all 20 amino acids, and with the highest content of arginine. And it's not only kernel of groundnut, but also the skin and the skin extracts, which also have a lot of antioxidants function. So therefore, it is a functional food ingredient and animal feed ingredient and also antimicrobial agent. So therefore, now groundnut is looked as a functional food, a complete functional food. And when it comes to breeding objectives, again, Dr. Krishna Prasad made a mention of the important focus in breeding of groundnut. One is the diseases like lately spot in Karnataka or in Kannada, we say tikka, and then you have the rust. And among the leaf spots, we have early leaf spot and late leaf spot. And then we have the sclerotium rot, aspergillus infection, bud necrosis, etc. And also there are several insects which cause major damage. And people are working on both conventional breeding as well as genomics-assisted breeding for insect pest resistance. And also the drought, high temperature, salinity, and iron deficiency, chlorosis, etc. are the abiotic stresses which cause a lot of damage and bring about the groundnut productivity. And in terms of nutrition, it is the oil, oleic acid, and other nutrients. As I mentioned, the resveratrol, a lot of efforts are going on in groundnut to improve resveratrol content. So these are the breeding objectives of groundnut. And when it comes to breeding, I'm talking of tapping of variability for breeding, whether it's for genomics breeding or even conventional breeding. Variability is very important. And in case of groundnut, the variability comes from its two subspecies of groundnut, because all of us know that groundnut originated from the Allah polyploidation. And groundnut is the Allah tetraploid with AABB genome, and A genome contributed by Arachis duranensis, and B genome contributed by Arachis hypiensis. And groundnut has got two subspecies. One is called as hypogaea, and another is fastigiata. And in case of hypogaea, we got two botanical types. One is called as hypogaea, and another is hirsuta. And in fastigiata, we have got four botanical types. One is Valencia, and is Spanish, Peruviana, and Equatoriana. And among these things, the most commonly cultivated groundnut comes from Valencia Spanish types. And these two subspecies mainly differ for the presence or absence of flowers on the main stem. Say, we all of us know that hypogaea types do not have the floral flowers on the main stem, and also they have got alternate branching, and the lack of, maybe they are long durated, and also they do not have any dormancy, seed dormancy. Whereas the fastigiae types have floral main stems. Main stems are having the flowers, and they have got the other type of branching pattern. And also another thing is they are short durated. So therefore, mostly the cultivated types are of Spanish types, which I told they have got a short breeding cycle. And also another unfortunate thing is they do not have seed dormancy. In certain cases, they cause a lot of problem. And groundnut Arachis genus has got almost 9 sections. And among them, the Arachis is a major section. And across these sections, we'll find almost 81 species. And most of these species, as you can see here, they have got different genomes. I'll come to that point later. And also, they have got, across these things, they differ for chromosome numbers also, starting from aneuploids of 18. And most of them have got 20 chromosomes because most of them are diploids. And only 2 species have got 40, the cultivated one and the primitive tetraploid, that's Arachis monticola. And these are the different genomes we find, A, B, C, D, E, K, F, H. So these are the different genomes, diploid genomes we find in the genus. And I'll put it in the as a summary. Groundnut species are grouped into 9 sections, and only those sections in Arachis will hybridize with the cultivated type, that's Arachis hypogaea, which is a allotetraploid with AABB genome. And Arachis has 81 wild species, and most of these species are diploids. And there are 2 aneuploids as well, and 2 are tetraploids. One is the cultivated Arachis hypogaea, and another is the primitive tetraploid, that's Arachis monticola. However, the introgression is difficult due to sterility barriers because, as I told you, most of them are diploids, whereas the cultivated is the tetraploid. So therefore, introgression is really difficult. But a lot of proposals and efforts have been made. In fact, these are the different pathways which were suggested by Simpson as early as 2001. And these are the potential options for transferring genes from wild Arachis to the cultigen. One is called as the hexaploid pathway, where a 2x or the diploid is crossed to a 4x type and cultivated type, and the 3x is the chromosome number of 3x is doubled to get the hexaploid, and that hexaploid is backcrossed to cultivated type to restore the tetraploids. Similarly, there is another approach called as diploid and tetraploid pathway, where the diploids are crossed and then doubling is made, and then to get the 4x, and that is crossed to the cultivated type. And in another pathway, which again involves diploid and tetraploid pathway, this is the thing which is done to get the 4x types. And a lot of suggestions and efforts were also made by A.K. Singh and his associates. And you can find these kind of approaches mentioned in ICRISAT newsletter as early as 1980. And of course, nowadays we've got a lot of molecular methods to insert or transfer the genes from the diploids, wild diploids, into the cultivated types, and which involves transformation protocols. And mostly in case of groundnut, you have got the standardized protocols for agrobacterial-mediated transformation, as well as other methods of gene transformation. And in vitro regeneration protocols have been standardized. A lot of genes have been cloned, and they have been transferred, and transgenics have been evaluated and characterized for various traits. It could be disease resistance, it could be drought tolerance, it could be quality, like that. And then the phenotypes have been studied at least at lab stage. And nowadays, in fact, we have been using gene silencing and genome editing also for improving groundnut. And to tap this variability from various diploids, which are present in the groundnut germplasm, a lot of interspecific derivatives have been developed. And these have provided as the better source of alleles for various traits. And there are lots of review papers. And also, I should make a mention of the efforts of Dr. Nalini Mallikarjuna of ICRISAT, who found out the 2N pollen, a pollen with the 2N status for generating the interspecific derivatives in groundnut. And with all these efforts, hundreds of interspecific derivatives have been developed in groundnut, and they have been employed for evaluation and also broadening the genetic base of groundnut. And that's why the interspecific derivatives have given a lot of variability. They have helped in creating a lot of variability, and also they have led to the varietal development. To name one, a success story in resistance breeding is GPBD4. The efforts to breed for GPBD4 started in 1991. In fact, the cross between KRG1 and CS16 was made when I was the MSc student with Dr. M.V.C. Gowda, who was my MSc chairman. He's the mentor. And this is derived from KRG1 and ICGV 86855. And this ICGV 86855 is an interspecific derivative derived from Arachis cardinacea, one of the diploids. And this GPBD4 is also known as Vikas, and probably this is the first lately spot resistant variety in the world, and released by Government of India for the states like Andhra Pradesh, Tamil Nadu, Maharashtra, West Bengal, and Orissa. And it is in great demand in North Karnataka, even after release of after almost 20 years. And now people are coming out with, say, different improved varieties over GPBD4. And apart from these interspecific derivatives, we also have a lot of synthetic amphidiploids. Again, I should thank Dr. Nalini Mallikarjun for developing many synthetic amphidiploids. In fact, she, through one of the DBT project, she has shared many of these synthetic amphidiploids, and which we used for breeding, integration breeding, as well as mapping. And also we made use of some of these interspecific synthetic amphidiploids and integration lines for mutation breeding and also for hybridization. So these are some of the synthetic amphidiploids which Dr. Nalini Mallikarjun developed at ICRISAT using different diploids. You can see here, starting from the original progenitors, that is Arachis duranensis and Ipiensis, and also she used Magna and other diploid genomes, including Buttigieg, K genome, all those things. So if you look at the pollen fertility, and these are the cultivated types with the pollen fertility of over 80%, and most of these synthetic amphidiploids with AABB genome also had very high pollen fertility. You can see over more than 90%. And these are some of the features of the wild or the synthetic amphidiploids. In fact, we are maintaining some of these synthetic amphidiploids for integration breeding. And these are the floral types. And how we made use of these synthetic amphidiploids for integration breeding and mapping is represented here. So this approach is called as ABQTL mapping, where we use the recurrent parents like ICGS 76, DH86, JL24, and ICGV 9114. This is a joint venture between US Dharwad and ICRISAT through a DBT project. And we used two synthetic amphidiploids, IS-ATGR 278-18 and IS-ATGR 5. And they have got B and K genome. B genome is contributed by Magna, and Buttigieg contributes the K genome. And we developed several introgression lines by backcross breeding, and this was also assisted by some of the markers which are listed here, especially for lately spot resistance and rust resistance. And we also made use of some of the SNP markers, and at the end, we could get some of the introgression lines, which we call as IL3 and IL4. And also these backcross lines or backcross populations were used for mapping. So therefore, this approach of ABQTL has got dual advantage. One is we can develop the introgression lines, and also simultaneously we can map the QTLs. And of course, this is one of the derivative of that project, where we subjected the IL3 and IL4 introgression lines for mutagenesis, and then we identified a couple of mutants. You can see here, this is a mutant of IL3, which is higher yielding than the IL3. And also it is highly resistant for not only for lately spot, but also for the ELS, early spot. And later at ICRISAT, Dr. Shivali Sharma developed several introgression lines again, like ICGL 17105, 17110, and 17112, like that. And we are fortunate to get those interspecific derivatives from her. And then again, we used these introgression lines because they were highly resistant for ELS and LLS. So therefore, we subjected those introgression lines for mutation. And also we used them as the donor of resistance with some of our own variety as the recurrent parent. And these are some of the improved mutants which we generated from these introgression lines. And also we are using them as a donor along with GPBD4 to improve the resistance as well as yield level of GPBD4. And here there are two lines which are derived from GPBD4 and 17112. And you can see the yield levels when compared to the GPBD4. GPBD4 has given almost 30 quintals, whereas these derived lines have got almost 36 quintals. So like this, the pedigree breeding, mutation breeding have also resulted in the development and release of a lot of varieties in groundnut. But the conventional breeding has its own challenges. And those challenges mainly come from the interactions among the diseases, linkage drag, and resistance is distributed in different germplasms, and transferring is really difficult. And also resistance is more of quantitative in nature. That has been very well established in various resistance breeding programs, and also significantly influenced by the environment. So therefore, we can say that the overall success rate of conventional breeding is little limited. So therefore, what we thought of the genomics-assisted breeding over the conventional breeding, because already people have addressed and witnessed the advantages of genomics-assisted breeding in terms of increasing the speed and efficiency of breeding, to select the disease-resistant QTLs, accelerate the recovery of recurrent parent during backcrossing, because of a lot of markers can be used for say checking the level of recurrent parent genome, and also substitute for phenotyping in the early phase of breeding, whether it is for marker-assisted breeding or even genomic selection, like that. And also there is a possibility of breaking the linkage drag, because we are looking at specific QTL, and we can ensure that only those QTLs are transferred with recombination selection. And pyramiding of different disease resistance genes can also be facilitated by the genomics-assisted breeding. And hereafter, I will talk of the main work which was carried out in our department under the mentorship of Dr. M.E. Gowda and Dr. Khadi. And we worked almost about 12 years on foliar diseases in groundnut. That is early leaf spot and late leaf spot. Early leaf spot is caused by Phaseryopsis, whereas late leaf spot is by Personata, and rust is caused by Puccinia arachidis. And these two diseases, that leaf spots and rust, when they co-occur, they can cause almost 70% of the yield loss. You can see here, this is the ticca and this is the rust. And the susceptible varieties where there is complete defoliation because of late leaf spot, that can result in almost 70% of the yield loss. And it does not only affects the yield loss, but also affects the say quality of the produce. In fact, they also predispose the soil to the other pathogens. And there is a lot of variation among the genotypes for the yield loss. See, there was a study conducted by Dr. Gowda in 1991 and 92. You can see the differences for the yield loss. Say, for example, here, there is a variety called DH330. Say during 1991, the yield loss was almost 45%, whereas in 1992 it was 27%. And same is the case with even KRG1. So there is a lot of variability among the genotypes for the yield loss as well as the resistance. And there are also chemical solutions available for controlling these diseases, like fungicides, but they are used very at a low level due to the uncertainty under rainfed conditions, and also therefore breeding for resistant genotypes is always better and economical. So in that sense, the genomics-assisted breeding was taken up in our lab along with the collaborations from ICRISAT as well as KDRI Japan and other institutes. Say, when we talk of genomics-assisted breeding, there are various components. The first component could be developing the mapping populations, association panels, and training populations. This is very, very important. And when you have good mapping populations, then the mapping of QTLs becomes very easy and it becomes more efficient also. And the second component could be developing and validating new marker systems. Because in case of groundnut, the problem is very low polymorphism. Say, though we find a lot of variability among the phenotypes, at DNA level, there is very low level of polymorphism. And with available markers like SSRs, RFLPs, and RAPDs, people have observed just 5% of the polymorphism. It indicates that if you use about 100 markers, then for mapping, only 5 markers are useful. And the next step could be genotyping and extensive phenotyping. This is again very important. When you have a lot of accurate phenotypic data, then our QTL mapping is more accurate. And then constructing the linkage maps and consensus maps, constructing the QTL maps, identifying the QTL regions, markers, and their effects, and marker validation. I would say this is another important stage. And then employing these markers for MABC and the marker selections, and then developing backcross and introgression lines, and developing the improved genotypes when we have the markers and the QTLs known for a given trait. So therefore we can say that we can come up with a product starting from the developing of mapping populations. So that's what we have done in case of groundnut over the last almost 10 to 12 years. We have taken various recurrent parents. Most important are the JL24 and TME2. And using this marker-assisted breeding, we could develop the products for the backcross lines of TME2 and JL24, which are resistant for lately spot. And these are the mapping populations which were already available at US Dharwad with the great contribution from Dr. M.U.C. Gowda. In fact, he had developed almost 11 mapping populations. And these mapping populations, again, they were used by different centers at ICRISAT as well as Junagadh. And then out of these mapping populations, the first two, that's TAG24 and GPBD4 and TG26 and GPBD4, which differed for lately spot and rust, they were extensively used for phenotyping and genotyping by ICRISAT. And also the population size was very good. In fact, in the first population, it was almost 268, and in the second population, it was 146. A lot of PhD students have also contributed, like Kedikar and Sujay and Sarvamangala and Varsha Kumari. And these mapping populations were extensively used for phenotyping as well as genotyping. And Dharwad is considered as an ideal location, especially for lately spot and rust. Say sometimes you go for artificial epiphytic conditions, otherwise even by nature also we get regularly the lately spot and rust regularly. So therefore evaluation for these diseases is very easy as well as accurate. And also we can see the kind of distribution of the members of the population, that's the real population. And the second important resource which is available for genomics-assisted breeding is the availability of the markers. Say, though there are several markers which are used in groundnut, like RFLP, SSRs, all those things, even DART markers, the problem is the less polymorphism. As I told you, it's just 4 to 5%. So therefore, we thought of developing new marker systems, like transposable marker systems, new SNPs, like that. So in case of groundnut, a lot of SNPs are already available because the genome sequences are available. Genome sequences of the A genome, that is Duranensis, A B genome, Ipensis, and Arachis monticola, Arachis hypogea, they have been sequenced, and also a lot of SNPs have been identified. And with respect to the population genomics, this was our first initiative where we published a chapter by using the whole genome resequencing data of 178 accessions, which comprised of cultivated tetraploids, wild diploids, synthetic amphidiploids, and they were compared with the reference genome of Tifrunner, which is a cultivated type. And then we identified important features of population genomics in peanut. And also recently we identified almost 43 lakh SNPs by analyzing almost 179 genotypes. And also we identified the structural and functional features of these SNPs. And for example, these are the chromosomal distributions of the SNPs. There is a lot of variation, you can see here. And the A genome has got almost 29 lakhs, whereas the B genome has got 23 lakhs, though B genome is a little larger than A genome. And these are the chromosomal densities for SNPs. A08 has got the highest SNP density, whereas the B genome, that is B01 chromosome, has got the lowest SNP density. And also we compared several genotypes. This is one synthetic amphidiploid, and when it was compared with the reference genome, we found the highest number of SNPs, whereas there is one US variety, there is a PI 628538, and this showed very less number of SNPs, indicating that this is much closer to the reference genome. And this is a very important graph, I would say. See, out of 179 genotypes, the level of SNPs, if you look at, then 50% of 43 lakh SNPs were identified when we used just 12 genotypes. Similarly, 99% of the SNPs were identified when we used just 106, say, genotypes. But for getting extra 1% of the SNPs, we required almost 70 genotypes, indicating that those 1% of the rare SNPs can be found only when we use a lot of extra genotypes. So this is a very good information in terms of SNPs. And also we checked what is the level of transitions, what is the level of transversions, all those things. And the rate of transitions was more than that of transitions across all the chromosomes. And this is also, again, the chromosomal distribution and also the distribution with respect to the genes. How many SNPs are located intergenic, how many are intronic, like that. And both intergenic and intronic SNPs were more than the exonic SNPs. And coming to the impact, okay, we classified the effect of SNPs as high, low, and moderate, and say modifier also. SNP impact was very high at 2,488 loci, and this was mainly because of the start codon lost or stop codon gained, and also the splice donor and splice acceptor changes. So this actually led to very high impact of these SNPs. And this information will be very useful when such SNPs are associated or linked to the traits. Then we can get the significance of these SNPs. And also we compared different genotypes for various traits using these SNPs. And also we worked out the polymorphic information content, which I told that the most of the markers in Groundnut, they have got very low peak value. However, when we analyzed about 43 lakh SNPs, we found that almost 46,000 SNPs had very high level of peak value. And I have to make a mention of another new marker system, which for the first time we developed in Groundnut. And that's called as the transposon-based markers, because we had a lot of interesting genotypes with us, again developed by Dr. M.U.C. Gowda. And one among them is called as Dharwad early runner. And we had a lot of derivatives of this DER, and they indicated a lot of high instability. So therefore, at that point of time in 1996, they thought that there could be a possibility of the activity of cryptic elements, that's the transposable elements. And these are some of the derivatives of DER. And these are the genetic instability phenomena which we observed in DER and its derivatives. And in 2008, the transposable elements were known in Groundnut, especially the miniature inverted repeat transposable elements, that's the MITE, through a study of Patel and his associates. And the insertion of this MITE element in HFAT2 led to very high olic acid content. And then we thought of identifying whether there is only one copy of MITE or there are multiple copies of this MITE. So therefore, we used the tail PCR to identify the exact location of this MITE element. And then based on that, we designed a primer, and we could track the activity of this MITE. And when we identified or when we checked the different genotypes derived from DER, we found that the MITE activity was highly associated with the high frequency origin of lately spot resistant mutants. So therefore, we could give a good connection between lately spot resistance and the transposal activity. And also we identified whether this MITE is activated by the mutations or somoclonal variations, gamma rays, like that. And in most of the cases, we found that it is not only EMS, but also the spontaneous mutants, somoclonal variants, and gamma ray mutants could activate the MITE activity. And also we could find the major difference between the two subspecies for the MITE activity. You can find here, most of the Fastigiata types had insertion of MITE at the given site, whereas the Hypogaea types had no mite in the that specified say site within the genome, and thus we could characterize the mite activity, and also we could give some explanation on the mutation and evolutionary origin of botanical types in groundnut. So what is apparent is H-MITE1 is activated by EMS, and H-MITE1 prefers the same site in 4 independent mutations, and also it could give some basis for the mutation and subspecies differentiation. And also this mite has got some specific insertion preference in terms of the genes. It mostly prefers the genic region for transposition, and therefore it seems to be a reliable marker. That being the case, we use this marker system for genotyping a lot of mapping populations which were available with us. And later, a scientist called Shirasawa from KDRA, Japan, he used the transposon display using this mite specific probe, and then also used in silico analysis. And using these two techniques, he developed almost 1,000 markers. And then when he checked the Japanese genotypes using this marker system, then he identified a polymorphism rate of 22%. See, you can see here the polymorphic rate of 4 to 5% with the remaining markers and the transposable element markers. And later, we used the WGRS data of almost 33 genotypes, where we had sequenced our own 6 genotypes like DER, VL1, 110, 110S, ICGV 86855, and VG9514. And apart from that, we also used the remaining 33 genotypes. And then we searched the entire genomes of these 33 genotypes for the mite element. And then we identified almost 2,957 sites where the mite was present, and their distribution across the genes was found out of which there were about 1,400 which were intergenic, about 81 in the exonic region, all those things. And these almost 3,000 markers have been developed. And now for the first time in groundnut, we got 3,000 AHT markers available for various applications. And we also tested across different mapping populations about their polymorphic rate. And you can see here, there are certain mapping populations which show almost 35% of polymorphism. On an average, across the various mapping populations, we could identify about 20% of polymorphism, which is almost 4-fold more than the routine markers like SSRs, RFLPs, or even RAPDs. And also, we could convert some of the SNP markers into CAPS markers. And we also could, because we had a lot of mutations and mutants, and we could provide some of the structural basis of structural mutations in terms of the copy number variations, transposable elements, and also the SNPs, and also we could find some of the somatic mutations. And these AHT markers were widely used for mapping, both linkage mapping, association mapping, and then background selections, studying the genome instability and rearrangements in our various studies. And QTL mapping, this is the one which we have done for lately spot and rust. And our first effort started with the collaboration between US Dharwad and ICRISAT. In fact, Khedikar, who was a PhD student of Dr. M.E.C. Gowda, went to ICRISAT with Dr. Rajiv Varshney, and he helped in genotyping. And this is the first study where we identified the genomic regions, that is linkage group 12 and linkage group 15, which contributed for lately spot and rust. You can see here the PVE, phenotypic variance explained, to the tune of 60 and 80, which is very, very high. And we rarely find such PVEs for any traits, and also very high LOD of 13 and 69. And that kind of mapping we continued with more and more phenotyping and usage of more markers like that. And this was the next study by Sujay. And then we also used my own student, Kolekar, also continued that kind of mapping. And in this case, we used the phenotypic data from 13 seasons, where the mapping populations were continuously evaluated for 13 seasons. And also she used a more number of markers in terms of SSRs, RAPDs, and transposable element markers. And again, we identified the same region for the contributing for lately spot and rust. And this is very important. This is the first study. You can see the genomic, it is almost a kind of a fine mapping, where the same region of the groundnut was inserted or identified with more and more number of markers over the years. And this region is from age 15. And this region later, when the diploid genome of groundnut was sequenced by Bertioli and his associates, they identified one R gene, NBS LLR gene, which is an R gene. And this R gene was found in the same QTL region, which we said that there is a QTL for lately spot and rust. And this actually supported our study and gave a lot of strength in going for validation of the markers using various approaches. Like for the first time, we developed a near isogenic lines from heterogeneous inbred families, and they were used for validation. And also we used different mapping populations, and all these validation works gave a lot of confidence that these markers are working for lately spot and rust, so that we can initiate a lot of breeding program. And also we undertook the QTL-SIC and genomoid association panel association mapping. This also led to the identification of the same genomic regions for lately spot and rust resistance. And this also went for fine mapping using various DDRAT sequencing and WGRS using different populations. And finally, we could fine tune starting from 8.8 MB region, I'm talking of physical region, to 2.7 MB region on A02 chromosome. And similarly, on A03 chromosome also, we boiled into 2.7 MB region, and on A02, 1.4 MB region. And also, we could identify some of the candidate genes in these two regions, which contribute for lately spot and rust resistance. And these were, in fact, the DNA pattern of DNA methylation was studied, especially among these 10 candidate genes. And also, the expression pattern of these 10 genes were also studied. And using all these genomic resources, we went for molecular breeding. And these were some of the recurrent parents like GL24, TME2, DH86, ICJ76. And then we took a lot of resistant material like GPPD4 and some of the interspecific derivatives like that. So a lot of marker assisted breeding was done using these markers for foreground selection as well as background selection. And we also selected TME2, and these are the reasons, because even after 60 years of release, TME2 is still popular among the farmers. Same is the case with GL24. It was released in 1978. Even now, in some of the areas, GL24 is cultivated, but both these varieties are mega varieties, but they are susceptible for lately spot and rust. And these are some of the markers which we used for foreground selection and background selection. And this is a background scheme which we used for backcross breeding. And using all these approaches and methods, we developed DBG3, which is a backcross line of GL24. You can see here in the field, the GL24 showing a lot of disease, whereas DBG3 showing less disease. Same is the case with the TME2 showing a lot of disease, and DBG4, a backcross line of TME2 being the resistant genotype or backcross line. And we also studied the stomatal opening in the resistant parents, donors, or recurrent parents and donors, as well as the backcross lines. There is a clear-cut difference in the stomatal opening. And these are some of the reactions you can see here. DBG3 versus GL24, always over the years, start from 2014 to 2021. The maximum score for LLS was almost 4 in term for DBG3, whereas GL24 it was 9, which indicates the susceptible reaction. Same is the case for DBG4 and TME2. And this is a yield level of, say, DBG3 and DBG4 over their recurrent parents. We got almost 30% in case of DBG3, whereas 47% in case of DBG4, higher yield than the recurrent parents. And this is the performance of DBG3 across the trials, station trials, multi-location trials, farm trials, and large-scale demonstrations. Finally, there was about 28% more yield than the GL24. And same was the case with DBG4, which is a backcross line of TME2. Across the different trials, there was about 29% more yield than the recurrent parent. And now the university has taken a decision last year to release these two backcross lines. In the DBG3 was released in the name of improved GL24, and DBG4 was released as super TME2. In fact, Dr. Kadi sir has already grown this DBG3 in his farm, and he's happy with the disease resistance in DBG3. And we also got the IC numbers from NBPGR, and I'm yet to propose before the state subcommittee, maybe next week. And such efforts are also reported from ICRISAT and other institutes in India. And ICRISAT has taken 3 different varieties, and those lines have completed various levels of testing. Probably this year we can expect the release of such varieties. And apart from this breeding for lately spot, we are also doing some kind of molecular breeding for high oleate content. So this is the scheme, breeding scheme, which we have been following. There is one special line called as Sun Olic 95R. It's an American variety, which has got almost 80% of olic acid content. Whereas most of the varieties which are grown in India, they have got about 40 to 45% of olic acid content. And increasing the olic acid content will lead to better nutrition as well as keeping quality of the oil or the groundnut. So therefore, we thought of using these Sunolic 95 as the donor, and GPBT4, G252, and other varieties, including DBG3 and DBG4, were used as a recurrent parent. And there are allele-specific PCRs available because there are two alleles or two loci like AHFAD2A coming from A genome and AHFAD2B, which are the homologous genes coming from B genome, are responsible for higher accumulation of olic acid content. And there are two mutations in these two alleles, and there are two specific PCRs available for checking and selecting those two mutant alleles. And after all these breeding schemes, we identified a couple of lines, backcross lines, which have got higher disease resistance as well as higher olic acid content. So these are some of the other activities in genomics-assisted breeding which we are doing. One is to identify the linked markers in biparental populations as well as magic populations. There is a magic population available from ICRISAT, and through NASF project, we were given the mapping population, and we have been using that mapping population for identifying the QTLs for drought and high temperature. And also in association with the ICRISAT and University of Edinburgh, we have got one DBT program that is on genomic selection. And in this case, we have already evaluated a training population for several years, and also several genome-based trait prediction models have been identified and developed, and these are being identified and used for selection in various breeding populations. And mapping of other traits, of course, worldwide, if you look at the important traits are the early leaf spots, late leaf spot, stem rot resistance, aflatoxins, tomato spotted wilt virus, bacterial wilt resistance, and resistant to aphid and rosette disease. All those things have been addressed, and a lot of genomics-assisted breeding and work is going on in this area for mapping as well as breeding. And apart from this, there are also other approaches, mainly the transgenics and involving genetic engineering, where genotype independent transformation protocols have been developed and various genes have been cloned and used in groundnut, especially the PR proteins from Solanum nigrum and Raphanus sativus. And when these two genes were expressed in groundnut, they led to resistance to late leaf spot. Similarly, there are other genes like chitinase genes. When they were expressed in groundnut, the transgenics were resistant for ELS, LLS, as well as rust. Similarly, the chitinase from rice was expressed in groundnut. Again, the resistance to LLS was observed, and defensin genes, and even people have used RNAi, gene silencing and RNA silencing for reducing the aflatoxin accumulation and aflatoxin control. And nowadays, people are also using RNAi for increasing the oil quality as well as oil content by depressing certain genes. And even for drought tolerance, people are using MIB repressors, Werke transcription factors for salt tolerance, and then DREB genes for soil moisture deficit stress and expression of MTID, that is a mannitol phosphate dehydrogenase for soil moisture deficit stress resistance. And then gene editing. People are using gene editing, both CRISPR-Cas as well as talents for various traits. And also you can find the speed breeding. There are a couple of efforts going on. So in conclusion, what I would say is groundnut has an unprecedented progress in genomics because of its sequencing, mainly the progenitors, A and B genome donors and cultivated genomes. A lot of efforts are going on in understanding genome now, and genome sequencing, marker development, and genomics-assisted breeding. Not only development of the resources, but these resources are being used for marker-assisted breeding to develop the products like that of DBG3 and DBG4, and also a few products from ICRISAT like Girnar 4 and 5. And also we are addressing the genomic selection, development of the markers, calculate the breeding value for the genotypes and the markers, so that the better genotypes can be selected based on the genotype, not based on the phenotype. And also implementing speed breeding, development of transgenics, gene editing, overall to improve the productivity of groundnut. So I should thank my funding agencies like DBT, ICR, DSTGSPS, DBT, BBSRC, and BRNS, RQA Project, GOK, my mentors, Dr. MEC Gowda and Dr. Khadi, and collaborators from KDRI Japan, Dr. Kenta Shirasawa, and people from ICRISAT, Dr. Rajiv Varshney, Dr. Manish Pandey, and co-PIs from Dharwad, Dr. H.L. Nadaf, Dr. Motgi, Dr. Lingraju, Dr. P.V. Patel. In fact, they helped a lot in a pathological aspect. Dr. Lingraju and P.V. Patel, being the pathologist, they could evaluate a lot of material. And in fact, my own students, PhD as well as MS students and field assistants, because all these years of almost 10 or to 12 years of field work involve a lot of field assistants. And finally, I should thank you for patient hearing. Thank you, sir.
Thank you very much, Dr. Ramesh S. Bhat. Really, it's a very thought-provoking lecture with elaborate research in both in traditional breeding as well as the genomics in groundnut breeding. Anything, Dr. Aparna? I want to any questions?
So there are questions in the chat box. You may like to read it.
Okay. Okay, so
Dr. Rajiv has shown his hand. Maybe you can ask him.
Sir, as usual, yeah.
Dr. Rajiv is shown his hands. You can ask him to.
Yeah, please carry on, doctor.
Hanji, sir. Okay, thanks, Dr. Tiku. Thank you very much, Dr. Bhat, for such a illuminating talk. My question is a very basic question regarding, it's only about the marker-assisted backcross MABC that you guys have been using, and going by the enthusiasm that you have shown with the basically all the applications of hello. Am I audible?
Yeah.
Am I audible? Yeah. So how successful has it been? And I mean, because hello, am I audible, sir?
Yeah.
You are,
okay.
please go ahead.
Yeah. I mean, using a limited number of markers, practically looking at the amount of the number of markers which a crop breeder would like to use, within a limit of, let's say, 300 to 1,000 or maybe 1,500. So where would you put? I mean, do you think it's really? I mean, it's this can really replace phenotyping as far as the recurrent parent is concerned, and can the breeder really rely on it? And would they be willing to rely on it with a reasonable amount of expenditure?
Yeah. Can I go ahead?
Please, sir.
Yeah. See, as of now, groundnut is very rich in terms of genomic resources, I would say. Earlier, of course, there were not many markers available, but now we have a lot of markers, and most of these markers are reliable. And also, see, the kind of mapping we have done in case of lately spot. I showed the phenotypic variance explained. It was to the tune of 60 to 80%, which almost clearly indicates it's an oligogene, right? And also, we got a lot of markers, and these markers are stable and reliable. So therefore, the marker-assisted backcross breeding has been very successful, at least for nematode tolerance in US, and then lately spot resistance with our own experience, as well as ICRISAT's experience and Junagadh, and also high oleic acid content, the high oleate trait. Again, the ICRISAT, and we are also doing, and nowadays I would say, or even like even in fact, several literatures also say the marker-assisted backcross breeding has become a routine in case of groundnut.
So how many markers would you put it? I mean, normally around how many markers do you think would be just about enough in
yeah, see,
MABC?
for one QTL, at least one marker.
Okay, yeah, and Dr. Bhat, you also indicated that the the genotypic polymorphism is a lot limited as compared to the phenotypic polymorphism, and you put a figure at something like 5%,
Yeah.
And now, obviously, there's a lot of hidden polymorphism which is not being detected by the genotyping. So, I mean, doesn't it have ramifications? When we are doing genotyping, how can a breeder really rely? When we put both of these data, both of these observations together. So,
Yeah.
I mean, do we observe this in MABC for the recurrent parent selection also?
Yeah, definitely it should be possible. See, rather than other types of markers, now we can really depend on SNPs. There are, say, at least we have identified 43 lakh SNPs, and out of which almost 50,000 have got very high polymorphic information content. And nowadays, there are various chips available, and genotyping is really easy. And in fact, ICRISAT already has developed a chip where thousands of genotypes can be very easily screened or genotyped, and that data can be used. And therefore, we are using this scheme of evaluation even for genomic selection, where our objective is to select the genotypes not by phenotyping, but by genotyping.
Thank you very much, Doc. That's very clear. Thanks a lot, indeed.
Yeah. Any more questions, please? Otherwise, I will summarize these.
There is Sameer Joshi raising the hand.
Okay.
Sameer?
Dr. Kadi sir also, I think.
Yeah, thanks.
Yeah, Kadi sir is also.
It was a first of all compliment from my side.
Dr. Sameer is.
Yeah, it's first of all compliment from my side. It's a very nice and comprehensive talk. My question is, in your talk you mentioned about along with the genotyping, phenotyping plays an important role. Are you using any precision-based phenotyping methods, or if not, have you thought about using any of them in future?
For genotyping, sir?
No, for phenotype. For
phenotype, yeah. See, always we have used because we have been working on lately spot and rust. And Dharwad is an ideal, location for getting the lately spot and rust naturally. In fact, we go for artificial inoculum also, but most of the season, in both the seasons, we get very good development of the disease. So therefore, we would say that phenotyping is most accurate in our case. So therefore, we could identify the QTLs with a lot of or very high PVE and with very high lot, and they have been reliable and they could be validated across the genotypes. So therefore, we could use them for marker backcross breeding.
Hello.
Anisa, please.
Excellent presentation, Dr. Ramesh, and also excellent work done by you on Groundnut, especially during my period. I was just looking for, and after the retirement, I could grow one of his varieties, that is DBZ3. Excellent, completely free from the disease. Along with the local, I had put the local was completely devastated, and this variety was completely free.
Yeah.
And very good contribution, and 2-3 varieties I think he is having at present.
Yeah.
I think all those varieties should be released for the benefit of the farming community, especially for this region of the state. That's what I feel. I once again congratulate Dr. Ramesh for having done excellent work and excellent contribution to the groundnut farmers. Thank you.
Thank you for your support, sir.
Very nice. Anybody?
So there are a few questions in the chat box. I'll read. Mr. Danish is asking, why stomatal study is required in your research study?
Sorry, what is?
Why stomatal study?
Oh, stomat. Okay, just to see the kind of stomatal opening, because that gives a kind of a mechanism of resistance. Okay. In fact, Dr. Lingraj was very crucial in doing that kind of work, just to see whether there is a difference between the resistant type and susceptible type. And fortunately, we found a lot of difference in the stomatal size of the stomatal opening.
Okay. Mr. Prashant Khadekar is asking, development of composite from mutant population is possible?
Composite?
Development of composite from mutant population.
Mutant population composite. In fact, I did not understand the question, first of all.
You can read it. It's written here.
Yeah.
Development of.
The meaning of composite mutant. I don't know. If you can explain.
Yeah, maybe Mr. Prashant, you can raise your question, raise your hand and ask the question by yourself. Mr. Prashant?
Yeah, he can explain. You can ask.
Yeah, please.
I think as I understand it, he is asking whether with various mutant populations you could then develop a composite variety.
Yeah,
I think,
exactly.
I think that's what he's asking.
Okay,
How
okay, okay.
He can
Yeah.
clarify. Yeah.
Okay. Say in groundnut, probably that is not a good idea because we have got a lot of good mutants for various traits, and we can go for a pure line variety instead of going for that one.
Yeah.
Mr. Anjan Gowda is asking,
Yeah.
since the PBP explain indicates that the casual segment is major gene, oligogene, I really want to understand why QTL mapping approach was followed instead of major gene mapping approach.
Yeah, that's a good question, in fact. But we never know unless we do it, right? Because in most of the cases, when you go for normal distribution on these things, though there were clear indications in the literature, also people have worked out the genetics of lately spot resistance earlier based on the segregation pattern. And in certain cases, depending on the genotypes, there were various reports indicating a single dominant gene, recessive gene, and multiple genes like that. But when we started the QTL mapping, we identified that the QTL region which we identified had a lot of FLOD and PVE, almost showing a oligogen pattern. But it doesn't mean that there is only one gene contributing for lately spot resistance, but the QTL region which we hit had a PVE of that much. But there are other minor genomic regions also. But for molecular breeding, markerist breeding, we concentrated only on that region which showed very high PVE. But from our mapping populations or mapping studies, we also identified some of the other genomic regions which had minor PVE. So I think that makes clear.
Okay.
Yeah.
Mr. Nimar Chandragiri
and in fact, in the QTL region, when we analyzed by fine mapping, as I showed you, there were 4 or 6 genes in the QTL region.
Sir, cloning of any genes has been performed, sir? No. These recent genes.
For lately spot resistance?
Yeah.
No. None of the genes have been cloned from this QTL region and tried for overexpression. But there are other genes from rice and other sources which have been put in or transferred to groundnut, and resistance to lately spot unrest have been observed by transgenic approach.
Mr. Nimar Chandragiri is asking, can you suggest any software tool for turmeric crop modeling?
Okay, for genomic selection?
No idea. Just
Okay.
asking.
Anyway, in most of these cases, I have been using R programming for various types of analysis, which can be used for the kind of analysis we are doing. And of course, for mapping all those things, cartographer and map makers, all those things are there. But the same thing can be done even using R programming.
Mr. Dinesh is asking, anyone working for providing DNA fingerprinting with SNPs or SSR marker?
DNA fingerprinting for? Sorry.
Fingerprinting with SNPs or SSR marker?
Right now, no. But we can think of that one in future based on the chips if they are made simple. But at least in the labs, they are available. We got 2 different types of chips with almost 40,000 SNPs and one with only 10 SNPs. And these 10 SNPs are for lately spot resistance as well as high oleic acid content. So if there is any line with disease resistance and high oleic acid content, this chip can be very easily used, and the lines can be identified very easily.
What is the best source of available here? Yashashwini Kulkarni is asking.
Yeah, what's the best source of?
What's
the best source?
No, it's not available. Can you please ask Dr. Kulkarni? Can you ask your question? It is not visible here.
Yeah.
Um, Mr. Yashaswini Varanasi is asking,
Mhm.
what is the best source available to learn our program for beginners?
Best source is, of course, Google. You start learning and see each and every package and functions. These things are available, but only thing is how best you practice on your own is very, very important. But to begin with, you can start with whatever is available on Google, and then you are the self learning is the best thing.
Yeah, that's true. And also, Yashaswini, we add you that ADPBR also organized our program for beginners. Keep
Yeah.
watching the website.
Yeah.
When it is available, you can join it.
Yeah. In fact, I did not have much idea on R programming till say at least COVID time, and I made the COVID time as the best time to learn R programming at home.
Wow, great.
Yeah. In fact, my physiological plant paper, physiological plantarum paper, where we analyzed 43 lakh SNPs, is purely done with R programming.
Mr. Abhishek is asking, what are the major phenotypic traits that are responsible for LLS resistance in groundnut?
Phenotypic, of course, the lesion number, lesion size, incubation time, they're called as the mechanisms of resistance, and they are very, very important. Lesion diameter, lesion size, lesion number. In fact, the score what we give contains all those parameters, most of those parameters.
Is there any other question? Anyone would like to raise the hand? If not, then Krishna Prasad sir, maybe you can conclude the session.
Thank you, madam. Thank you, madam. Doctor, just I have one simple query, not a question.
Yes,
you have mentioned about the fungal diseases. What about the peanut button virus disease, viral diseases like peanut button necrosis and stem necrosis? Some peanut, are they not harming or not causing?
No. Yeah, of course. No. They are very important diseases. At least not so important in Dharwad, but if you go to say Raichur region, it is very, very important.
Okay.
And also, we have got a mapping population which differ for that trait.
Okay.
Right now, because we are mainly engaged with the LLS and Rust, we couldn't pick up that trait.
Yes.
Yeah.
Okay. Nice. Thank you very much.
But we have a couple of mapping populations. If people want to use those mapping populations for mapping, definitely they are very useful.
Oh, very good.
Yeah.
Okay. Shall I? Okay, go ahead, ma'am. Shall I go ahead?
Yes, sir, please.
Yeah. Raju Taggar is something. Okay, nice. Yeah, this is a very, very interesting lecture by Dr. Ramesh S. Bhat. And nearly 68 participants have participated in this 1 hour program. It's a really record, I can say. And I simply conclude this. Dr. Bhat has explained very nicely the pathways to overcome the sterility barriers, especially since these species are having lots of problem with the sterility. And molecular methods to overcome the sterility barriers are also have been thoroughly discussed. And interspecific derivatives have been developed in the, it has been used. And through interspecific hybridization, they have developed about JPBD4. It is a source for the resistance breeding. And this has been released as a Vicos variety. It is very popular among the farming community. There are interspecific ampli-diploid population developed at Ikrishat having a high foliar fertility have been used in these programs. That was very good information. Introgression, breeding, and mapping from synthetic ampli-diploids, simultaneous mapping of QTL, and identification of mutants is one of the main things he has done in his entire research programs. ICGL lines from Ikrishat used for introgression lines through mutation and developed improved mutant lines, which have been widely used in his breeding program. Pedigree breeding and mutation breeding were successful, but it is a limited success. Now the genomic-assisted breeding with genomic tools nowadays, high level of technology tools which are applicable to the high people like the biotechnologists, not for the traditional breeders like us. And we never assisted in our lifetime in our during the period of our service. And genomic-assisted breeding steps, mapping population, validating new marker systems, genotyping and phenotyping, backcross and introgression. These are all the things have been done through genomic-assisted breeding. Population genomics of peanut explained is SNP distribution, that he has given a very good, clear, lucid presentation on SNP distribution. Impact of SNP analysis is very high. And new marker system, that is transposon-based markers, are being utilized for the high-level boric acid content. And AHTE, through AHTE, they developed about 3,000 markers available for screening the genotypes. That was the very good things they have made. Late leaf spot and rust resistance are the main diseases. He has made a tremendous progress in his breeding populations. Backcross breeding, through backcross breeding, they developed DBG3, that is a resistant, and the JL24 is a susceptible high variety. Utilizing those three things, they developed a very good lines. And DBG3 and DBG4, which has given a higher yields of nearly 2,039 kilos per hectare. And they released it as a improved JL24. And DBG4, they released it as a super JMBT release. And molecular breeding, high folic acid content, Sonolic 95R and ICGV 15025 used as a donor parents. They developed a high drought resistance, high folic acid content have been identified, developed and also identified for the further breeding program. They also initiated this multi-parent advanced generation interprocess, that is magic populations, transgenics, gene editing, CRISPR-Cas, speed breeding. These are all the things he has covered, almost all the things. It's a great talk. I'm very much, I learned a lot from his talk, and many of the people must have been learned a lot his talk. Thank you very much. Nice talk. And also thanks to Aparna Tiwari for giving me an opportunity to act as a moderator. Thank you very much.
Thank you. Thank you so much.
Thank you, sir. Thank you very much. Yes, Bhat, sir.
Yeah. First of all, I would like to thank again Dr. Krishna Prasad for your remarks, the organizers, ATPBR, all the members, and thanks to all the participants and those who have asked the questions also. Thank you. Thank you very much.
Thank you, sir. Thank you very much for a wonderful presentation. Definitely crop like groundnut, we don't hear it more often. So it's really a, enlightenment for everybody to know a new aspect of groundnut breeding. Thank you very much for giving us opportunity to know more about this crop. And I'm sure the people who didn't ask the question, if you have any query, Dr. Bhat is easily available on your questions and queries, so you can raise them. And thank you, sir. Thank you for taking out your time and giving us a prospect on the groundnut breeding.
I have just one point, because if I don't ask that question, I will not get sleep. As I mentioned in my introduction, if you go to US or Europe and all the things, groundnuts are very, very big size.
Oh, yeah.
And why we are not getting in India? Are they genetically different, or because long day or sing source? What is the reason? That
you're right, sir. In fact, when I visited Japan a couple of times, I always saw those bold types.
Bold types, big size.
In fact, they are double the size of Indian kernels.
Exactly.
Or even 3 times more than that.
Yeah.
But there is a good initiation at our Eclipse Center, Dr. Motgi, who is the groundnut breeder here, and he has initiated that kind of breeding. But definitely the test weight and this one size, if you look at, then definitely they cannot be compared to the ones found in US or Japan. But definitely there are a couple of varieties which are already available, and he is trying to improve the size with his breeding work.
Yes, sir.
That's a major focus in his breeding program.
Okay, sir. Thank you very much.
Now you can get a good sleep, Prasad, sir.
Yeah.
Don't ask her that question.
Yes, he's right. Yeah.
Okay.
And we don't find that small kernel types in US or Japan, at least.
Yes.
I have not seen in Japan.
Exactly correct.
They're all a big size. Yeah.
Right, sir.
Yeah. Thank you.
Thank you, Prasad, sir, for giving a wonderful moderation and making a smooth execution of the whole session. Thank you very much for your time. And thank you all the participants for your active involvement during the session. And I'm sure we will look forward to see you again next session, next month, with another exciting lecture. Thank you very much to all of you, and have a wonderful weekend. Have a good day to all.
Thank you.
Thank you, madam. Thank you, Dr. Bhat. Thank you. All participants.